Hi everyone, I'm Jake Joy. I am the reference lab manager here in Gainesville, Florida. I've been with Lifestyle for about 19 years. I got my SBB in 2014, have been doing supervisory and management things since then. For this presentation, basically, here come the leftovers. These are some minor blood groups. They're not the big ones that you encounter every day. Uh, so there's a lot of information coming up in this presentation. So don't get bogged down in the minutia. Focus on the main points. Most of these are relevant to an IRL and for difficult antibody IDs and patient workups. Uh, but you'll probably see some of these, especially the famous ones, on the exam. So without further ado, uh, here we go. And here they are. Uh, except for Lutheran, most of these are just interesting and fun. Some of these will be, some antibodies are benign, but some of these are deadly. Most of them you'll never hear of again. Um, they're rarely encountered, even in an IRL. A lot of these, except for Lutheran, we encounter Lutheran antibodies rarely, but Dombrock comes up every once in a while. Um, we see Chido Rogers, all these HTLA types, which we'll explain all that soon. Uh, they're not the most common things. You don't see them all the time, but like I said, they may show up on your ASCP BB exam. That's a pretty thorough exam, so let's get ready. Okay, the Lutheran blood group system. This is the sort of the biggest system that we're going to deal with in this presentation. A lot's known. It's been known for a long time. So, a fun fact, actually, this was discovered in 1945 in a multiply transfused patient that had 5 antibodies, and actually the patient name was not Lutheran, it was Lutheran, but in a misspelling, we ended up with Lutheran, and it stuck. So sort of an interesting fun fact there. So the function of the Lutheran antigens on the cell is basically cell adhesion and identification. The IGSF domains, that's the immunoglobulin superfamily of adhesion molecules. Uh, there are lots of antigens in this system, but really the most important, really all we're ever going to focus on are Lutheran A and Lutheran B, and to some extent Lutheran 3. The Lutheran A and B are, as you can see, antithetical codominant. So you can express both. So you can be both Lutheran A and B positive at the same time, or negative for one. You almost never see anybody that's uh, negative for Lutheran B. It's super high incidence. When you do, it's going to be that's going to start to cause a lot of difficulties in your workup. Lutheran 3 antigen is present basically if you have either Lutheran A or Lutheran B, you are positive for LU3. The only way to develop an anti-LU3 is to have a null phenotype, a truly you have no Lutheran A or Lutheran B on your cells, in which case you will develop a pan-Lutheran, and of course clearly that's going to cause a lot of difficulty for finding acceptable units for transfusion. Yeah, so the best way to think of an anti-Lutheran 3, it's an inseparable seeming mixture of anti-Lutheran A and anti-Lutheran B, but it's not separable. It's a one antibody that yeah, if you think back to RH, it's kind of like anti-G. It reacts with a couple of different antigens, but you can't separate those into 2 separate antibodies. It is a one antibody. So think of it as like a pan-Lutheran or a Lutheran total antibody. As you can see, these antigens are not well developed at birth, which ding ding, that should give you a tip for antibody IDs. If you've got access to cord cells, especially if you've made a high incidence, if you've got anti-Lutheran 3, or your Lutheran B, you may be able to use cord cells in your antibody ID, and that will help narrow down what system we're working with. Another interesting fact about the Lutheran system is that in 1951 it was reported that there was some linkage between the Lutheran system and Lewis. It actually wasn't Lewis, because if you'll remember from that system, it's actually the secretor gene that there's linkage with. So Lewis and secretor are closely linked, not linked in the genetic sense, but they depend on each other. But there's actual genetic linkage because of the proximity of Lutheran and secretor on the chromosome. Something interesting about the Lutheran blood group system is you actually have 3 different ways for a null phenotype. Now, null phenotype as in when you type these cells, they're going to appear to be negative for both Lutheran A and Lutheran B. However, only one, the recessive genes, basically it's that silent allele, and if you get the silent allele homozygous, then you are truly there is no Lutheran substance on your cells, and you are also Lutheran 3 negative, can make that anti-Lutheran 3. The other two versions, you won't make an anti-Lutheran 3. There are trace amounts. The inhibitor, the NLU, that one, that is a dominant gene that will regulate the expression. Actually, you can only get the heterozygous version of this if you develop the homozygous version, then that is not compatible with life. So you won't encounter a person that is homozygous for this, because they won't survive. But basically, you still get very, very weak expression of your Lutheran antigens, and as you can see, a couple of others. The other X-linked recessive inhibitor is a similar situation. You're going to make very, very trace amounts of the antigen. You won't be able to detect these by typing, but if were you to do adsorption or elution studies, you can detect that there is some trace amount of Lutheran antigen on these cells. And of course, if there's any substance present, then that's why you're not going to make an anti-Lutheran 3 in either of these second 2 cases. Take a look at these antigen frequencies. You can see that Lutheran B, very, very high incidence. Most all of your panel cells are always going to be Lutheran B positive. You may or may not get a Lutheran A positive cell on a panel. Of course, if you have a panel with a single reaction and it's Lutheran A positive, then gosh, that should really that's an easy one. You just need to find a couple more Lutheran A positive cells to confirm it. Now, if you have a pan-reactive antibody ID panel, then probably your next step would be to test some cells with chemicals and enzymes. So if you end up doing that and you have a panel, or your cells are negative with DTT but remain positive with Ficin, that can steer us in the direction of maybe you have a high incidence anti-Lutheran B. So my next step then would be to look for some panel cells. Maybe they're expired, but maybe you happen to have one on hand that is Lutheran B negative. If that's the case, test that one. If it's negative, now we're really focusing on maybe you've got an anti-Lutheran B. Also remember those cord cells. if you were to test the same panel in the same technique, and your reaction strengths go away or were significantly weaker, that is another indicator that maybe we've got something in the Lutheran system. I would definitely try both of those techniques before moving on to your super rare frozen cells. Those are really hard to come by, and we don't want to thaw those out without good reason. That's kind of your last step to confirm your suspicions of a high incidence antibody. The Lutheran antibodies. So there's three that we want to talk about. The rest of the system is they're not of interest really to us at all. So anti-Lutheran A, this is pretty rarely encountered. The neat thing about it is that it will often be a room temp direct agglutinating antibody. It's got a mixed field appearance sometimes, so that if you get a reaction like that, that might be that can help figure out what we're looking at. You're going to rarely encounter this. It does not appear on an antibody screen panel for the most part, and then on a full panel, maybe you get a Lutheran A positive cell. So it's possible that this antibody can fly under the radar. Good news is it's not really that clinically significant. Now, anti-Lutheran B, we kind of care about this one. It can cause problems. It's also very difficult to complete the antibody ID. Anything that is underlying, if you have other antibodies, you've got to got to deal with your anti-Lutheran B in order to figure out what is going on with the rest of their picture. So this can cause us some headaches. The anti-Lutheran 3, well, that one's going to be that's very interesting. It's a rare antibody to ever encounter, but just be aware that it does possibly exist. If you do end up with an anti-Lutheran 3, since it is a inseparable mixture, it looks like an anti-Lutheran A and Lutheran B together. You could perform an elution adsorption study. So you could adsorb it onto a Lutheran B positive cell, elute the antibody off of that cell, and then test it against Lutheran A positive cells, and lo and behold, it will also react with the Lutheran A positive cells. That's the interesting point of those inseparable mixtures. It's actually an antibody against all Lutheran, not just a combination of some anti-Lutheran B and some anti-Lutheran A. Gerbich. Okay, this is a pretty big antigen system or blood group system. There's a lot of antigens, but we're really only concerned with Gerbich 2, 3, and 4. Those are the high incidence, and those are the ones that will cause us some problems with antibody ID and can be pretty difficult to deal with. The Gerbich product it's important for maintaining RBC shape. The glycophorin C and D are, despite their name, not very similar to GPA and GPB, the ones from the MNS system. So they, it's a glycophorin, but they're not closely related. The Gerbich system, those antigens are also a contact point and antigen that Plasmodium falciparum it allows it to get into the cell, which, of course, that is the most deadly form of malaria. So thanks, Gerbich. Gerbich negative phenotypes are pretty rare. There's a few different versions. The one that's most interesting is actually the true null. You're negative for Gerbich 2, 3, and 4. In this case, you'll have hereditary elliptocytosis. You'll get, instead of your traditional biconcave RBC, they end up being elliptical, which you can see in the picture here. the glycophorin C helps hold the protein band 4.1 to the RBC membrane, which is integral for keeping the normal RBC, our little donut shape that we're so used to seeing. So if you get a Gerbich null, then your cells they don't know how to shape themselves, and they end up being slightly deformed. We don't see Gerbich antibodies very often. Fortunately, they're pretty rare because the finding antigen-negative blood is difficult. These antigens develop early in RBC development. I mean, before actually a lot of other antigens, which this makes sense because that GPC helps to anchor band 4.1. Which gives your RBC shape. So without it, the RBC won't develop appropriately. Of course, this means that it's capable of causing severe HDFN in cases that where it's present. It's usually an immune-stimulated antibody. So if you got somebody that's negative for a Gerbich antigen, multiple transfusions or pregnancies can result in a clinically significant antibody. The Diego blood group system. Now this one's really interesting to me. The protein that the system is based on, band 3, is a huge protein. It's really important for RBC membrane integrity. This is actually a really highly conserved protein evolutionarily. It's ubiquitous in all vertebrates. You find it on RBCs, and you also find it on kidneys. Interestingly, this makes up about 20% of all RBC membrane proteins. As you can imagine, any mutations or oddities with band 3 will cause RBC deformation. You end up with spherocytosis or ovalocytosis. You get a misshapen RBC if you've got a problem with band 3 and the Diego system. The main antigens in this system are Diego A and Diego B, and right A and right B. With Diego B, you find that in everyone worldwide, very high incidence. Diego A is only found really in people of the Eastern Asian descent, which of course then is also the indigenous peoples of America, which is interesting because it serves as more evidence of the Bering land bridge migration. There's only one amino acid change between Diego A and Diego B, and Diego A being low incidence. It was first described in Venezuela in the 50s after a severe case of HDFN. That child actually died 3 days after birth. So even though it's low incidence in certain parts of the world, anti-Diego A can be a problem. It can slip through traditional testing and cause problems, even though generally the antibodies are mild, but in some cases they can be severe and deadly. Right A is a relatively common antibody, even though we seldom detect it just because it's so low incidence. It's usually not severe, and when you get a patient with a case of with anti-right A, cross-match for compatibility is your best option. The XG blood group system. This one is not much of a concern in transfusion medicine. Antibodies aren't particularly potent. The antigens aren't very immunogenic. But it is interesting because of its heredity pattern, which we'll look at that on the next slide. And the XG1 gene is our focus. It's got 2 alleles, XGA and the XG silent allele. So because this is located on the X chromosome, it's got a pretty cool inheritance pattern for a blood group. The antibody is pretty rare. You're not likely to see one, but this does appear on most antibody panels, all the way out there at the end, usually on the far right. Despite its rarity. Yeah, you're not likely to encounter one of these, but just in case you do, it will be interesting. Cyana is another one that you don't see very often. This is another member of the IGSF. Remember Lutheran? It's a similar sort of protein. Its function is unknown. It's a transmembrane adhesion receptor protein, which is a pretty vague description. But SC1 and SC2 are the ones that are most commonly encountered when they are, and they're really the focus. The rest of them very, very rare, and you may never hear of them again. The antibodies are not generally clinically significant. If you have you see chemical treatment here may be helpful. The antigen expression may be reduced. It's not consistent, but pretty much they're resistant to most everything. They're well expressed on cord cells, so your antibody ID is starting to sound pretty difficult if you do encounter one of these. Sibling studies may be useful. Molecular testing, you may have to dip into your rare cells, but fortunately we don't encounter this often, and it's not clinically significant. So while antibody ID may be a challenge, it's also probably not that significant from a transfusion medicine viewpoint. This brings us to the Cartwright blood group system. This one is abbreviated YT, but everybody, we call it Cartwright. YT is an older name for the gene that produces acetylcholinesterase, which is this antigen is actually found on the acetylcholinesterase glycoprotein. You may recall that is its purpose, its function is to terminate nerve impulse transmission. So its presence on RBCs is a little interesting. It's actually unknown. We know that it's not found on white blood cells. There is no null phenotype in this blood group system, probably for obvious reasons. You would encounter significant difficulty in the function of your nervous system without this gene. You just imagine a sprayed roach. Those acetylcholinesterase inhibitors are a class of insecticide that we're also fond of. Cartwright antibodies are generally not clinically significant. We do see requests for antigen negative blood come in to the reference lab sometimes. It's a really tough antigen to find, so we freeze these rare cells when we do find them. You don't necessarily need antigen negative blood for a transfusion, although if you don't provide it, then you will have an incompatible cross match. Dombrach. This system's a pretty big deal. We deal with it a lot in the reference lab, at least providing antigen negative units. The function of this of the gene product is not actually known. It's an enzyme. It comes from the ART4 gene, which is the ecto-ADP ribosyl transferase 4. So we kind of know what it makes, but its function is actually not known. Of the 10 antigens, DOA and DOB are sort of the most common, but Holly, Joseph, and Gregory are some that we're interested in a reference setting. The Dombrach antigens are sort of your classic example of how enzyme and chemical treatment can help solve an antibody ID. The antigens are all resistant to Ficin, but sensitive to DTT, so that should sort of trigger Dombrach when you see an antibody, especially a high incidence antibody that reacts that way. Antigens in the Dombrach system are not very immunogenic. Mostly you see these in patients that are multiply transfused. Think sickle cell population, and of course the Holly and Joseph antigens are more frequently negative in the African American population, that can then start to present challenges when you have sickle patients that develop an antibody towards one of these antigens. Dombrach does have a null phenotype, and it's known as Gregory, and GYA, that patient will be negative for all Dombrach antigens. You can see that Dombrach antibodies have varying degrees of severity. The clinical significance does vary quite a bit among antigens in the system. DOA and DOB are sort of the most important. Because of the poor immunogenicity of these antigens, it really takes multiple exposures in order to make an antibody. So you frequently see Dombrach antibodies in combination with other antibodies. You don't see them as a standalone single specificity very often. Colton blood group system. This is one that you don't encounter very often, or at least antibodies to Colton antigens. This is kind of a neat protein. It makes multiple passes through the membrane and creates a channel. It's a water channel. Hence the name aquaporin. The three alleles are Colton A, Colton B, and just CO. You can make an anti-CO3 if you're negative, if you don't possess either Colton A or Colton B, if you have the silent allele for both. Colton A is really high incidence here. Colton B is low-ish. It's only 10%. Colton 3 is present if you have either Colton A or Colton B. Then there is a shared Colton 3 specificity. You have to be the null phenotype in order to make an anti-Colton 3, and it will appear as an inseparable mixture of anti-COA and anti-COB. These are IgG antibodies reactive at IAT, and they have been implicated in severe reactions. So a clinically significant anti-Colton 3 is going to cause some real issues with finding compatible, safe blood for transfusion. The Kromer blood group system is a large system with a bunch of antigens that you will probably never encounter antibodies to. It's on the decay accelerating factor protein CD55. It helps to tone down complement so that it makes your red cells a little less resistant to the complement system. And once you follow that to the end and get into the membrane attack complex, bad things tend to happen to RBCs. CD55 is also a receptor for Plasmodium falciparum. Kromer has a null phenotype. The NAb phenotype it lacks all Kromer antigens. Fortunately, there's another complement regulatory proteins, CD59, that can pick up the slack because spontaneous hemolysis of RBCs is generally not a good thing. Kromer antibodies are usually not clinically significant. We don't see Kromer antibodies very often in the reference lab. If one is suspected, then you can perform inhibition studies with either serum or concentrated urine from antigen-positive individuals. VEL. VEL is one that it seems like everyone has heard of, but is seldom actually seen. I think that sometimes there's confusion over the actual VEL blood group system and the V antigen that is found in the RH system. So please keep that in mind. The V antigen in RH is a totally separate thing than the VEL antigen. There's a single antigen here in this system. There's really high incidence, very hard to find antigen negative blood. Anti-vel can be very bad news. This may be why it seems like everyone has heard of it. It's a high incidence antibody that can be really deadly. It's been reported to be an autoantibody, but usually immune stimulated. There could be an IgM component, but this will activate complement and actively hemolyze RBCs. The SID blood group system used to be known as the SDA antigen. It was classified as in a series as a single antigen, but once the underlying genetics were understood, it was able to be reassigned to its own blood group system, and this is the only antigen in the system. Antigen expression for SID is all over the place. Some cells have got a lot, and then some are basically undetectable. You can find the antigen present in a lot of different secretions, but its highest concentration is found in the urine, and that's actually one of the techniques we can use for antibody ID. Is we can inhibit antibody activity with guinea pig urine. Anti-SDA is generally not clinically significant. It's frequently found as naturally occurring. About half of the people that are truly SDA negative will form a naturally occurring anti-SDA. Probably the coolest and most memorable thing about the SID blood group system is the appearance of the agglutinates under a microscope. Once you see it, you'll probably never forget it. It's pretty striking. The agglutinates they're refractile, and they're for lack of a better word, they're shiny. They shimmer. They have a really neat visual appearance, and I believe I've only ever seen it once, and it was striking, and I remember it. The antibodies formerly known as high titer low avidity antibodies, or HTLAs. That's no longer an acceptable real classification, although you will still hear people say HTLA, although many times you will hear them say HTLA-like, because they know that that classification is no longer acceptable. But these do all share a reactivity in common, and they have some similarities that we'll see on the next slides that we can see why they were sort of considered and grouped together, even though that grouping is no longer valid. Most of these have been assigned to their own blood group systems. And these characteristics are what brought those antigens and antibodies together. They all have sort of weak, tenuous reactions that many times are not reproducible. I can tube test a panel and get a certain number of cells reactive, and then the next person come along and see something significantly different than what I did. I could then go back and retest it and not even get the same results I did the first time. Agglutination is generally very weak with these antibodies, despite their high titer. If you were to do dilutions, you see that there is a high concentration of the antibody. They don't bind very well. And even more frustrating and confounding is that most of these are high incidence antigens, so they're antibodies. They can give you some trouble during antibody ID because of the way that they react and their hit or miss pattern. These can cause you to pull your hair out, but most of the time they're not clinically significant, and we have ways of dealing with them. Chido-Rogers is probably the most common, most famous of the antibodies formerly known as HTLA. These are actually not produced by the RBCs, but they absorb onto the surface of the RBC. It's actually a complement component. We don't need to find antigen negative blood for transfusion, just cross-match for compatibility, which of course can be frustrating sometimes if you get variable reactivity. Neutralization is the go-to method for neutralizing, getting rid of Chido-Rogers reactivity. They're also destroyed by enzymes, but not DTT, which is a handy tip for antibody identification. The NOPS blood group system is another one of these HTLA-like antibodies that we encounter semi-regularly. We don't, in the reference lab, you don't try to get as specific as NOPS A or McCoy or SLA. Once we have done our testing with Physin and DTT, basically we want to make sure that it's not another antibody, say a Dombrock, which would have similar reactivity. But once we've ruled that out, we can be sure that we've found something in the NOPS blood group system. But that's about as far as we go with identification. The antibodies are generally not clinically significant, so we'll cross-match for compatibility or transfuse carefully. Another one that has been assigned to its own blood group system is JMH. This is the John Milton Hagen blood group system. A handy tip to remember this one is because the antigen expression decreases with age. We once the antigen fades, you're more likely to make an antibody. So we see that antibody in older patients. And the handy tip is that John Milton Hagen just kind of sounds like an old man's name. So that always I use that as a tip to help me remember that fact about JMH blood group system. Another antigen that was formerly grouped into that HTLA category is actually not even a blood group system or a series or collection. The BG or Bennett Goodspeed is actually a white cell antigen that is present on some mature RBCs. On an antibody panel, you might see HLA out in parentheses at the side. That could be referring to the BG antigen. The 700 series is where we put antigens that don't qualify as blood group systems. They're very, very low frequency, and until they are understood and have their underlying genetics worked out, and we determine what the actual gene product is, you can't classify it as a blood group system. So super low incidence antigens and antibodies end up finding themselves in the 700 series until they can be reclassified. The serologic investigation of low incidence antibodies generally doesn't have to be very in depth. The practical value of really trying to lock down the specificity of one of these antibodies is fairly low. It can be fun to do and neat to actually put a pin in it, but most of the time you just need to select another unit to cross match. When you discover these, most likely it is upon cross match. You've done an antibody ID and you've selected antigen negative blood for the patient, and then you go to cross match it, and lo and behold, it turns out to be incompatible. You may cross match several units and all of them are compatible, and there's one that is incompatible, and most of the time that's due to an antibody directed at a low incidence antigen that unit happened to actually be positive for. If you do decide to investigate, then the normal steps of let's do some enzyme treatment, chemical treatment. Some places have a selected cell panel that are positive for various common low incidence antigens. Some history on the patient or even the donor unit that you are cross matching may also be helpful. It can guide you in the right direction to find find what low incidence antigen might be the culprit here. But for the most part, lows are easy. You just pick another unit, you go from there. The transfusion medicine implications here are very minor. The 901 series is sort of the opposite of the 700s. These are the really high incidence antigens. There's actually not many left. Anton is a good one. That one's actually got a reputation for being kind of bad news. Antibody ID here can be very, very difficult. The finding compatible blood can be even more difficult. This is where things get very complicated. You may have to seek help from the rare donor registry, even doing sibling studies and finding family members to donate blood. You might have to go that far to be able to provide compatible units to a patient like this. Serologic investigation of high incidence antibodies can be a daunting task. Unlike the lows where you just pick another unit for a transfusion, highs, we really need to figure out what we've got going on. Is it clinically significant? We need to do everything we can to identify this antibody, make sure that it's not a warm autoantibody, and then figure out how we are going to provide antigen negative blood. Determining that you don't have a warm auto is part of the recognizing that you've got a high. If you have a negative auto control and a negative DAT, then now we're not thinking warm auto. We're probably looking at either a combination of multiple antibodies or a single specificity towards directed at a high incidence antigen. If all of your reactions are of similar strength, then we should start to be really suspicious that we've got an antibody towards a high incidence antigen. If it's variable, like you've got some 1+ reactions and some 3+ reactions, well, that seems like maybe you've got a combination of different antibodies. But if you're 2+ across the board or 3+ across the board with a negative autocontrol and negative DAT, it really seems like we're in the high incidence territory. Once you've recognized it, probably the next step is going to be enzymes and chemicals. That's always a really good first step. See what the reactivity looks like. Then phenotyping. It could be something really easy. It could be an anti-little k or an anti-U. That simple serologic phenotyping would be enough to point you in the right direction. Patient history can also be useful. You know, what's their ethnic background, the family history. Perhaps they're a close family and they're consanguineous. We can also perform adsorptions, inhibitions, dilution studies, kind of the whole 9 yards. This is a case where we're going to kind of throw everything at it. Finally, once we get pointed in the right direction based on our other work, we will use a selected cell panel that's negative for some high incidence antigens to either confirm our suspicions or even to help us try to discover what's going on. These are difficult workups that can sometimes take many days, especially if we're waiting on a genotype to come back. From a transfusion medicine viewpoint, this is about as tough as it gets. Never underestimate the value and the power of enzymes and chemical treatment. I keep a table like this in my office when I'm reviewing antibody workups, and I consult it all the time. So you can just kind of look across the board here and check out the example. So if you do your testing and your reactivity remains after Ficin, but it goes away after you've done DTT treatment, that can start to make us think, gosh, it might be one of these. It might be a KEL antibody or a Dombrock or an LW. Now it doesn't narrow you down into the exact specificity, but it gets you into the system. And once you're into the blood group system, then we can start picking our selected cells and approaching the antibody ID from a little more confined and restrained approach. And this is a very, very useful table for helping to narrow down and figure out what it is you're dealing with, especially with a really high incidence antibody. And finally, sort of the culmination of all this, and the most important thing, is how do we find compatible blood for a patient? For lows, we've talked about this. It's easy. You just pick another unit. Now the highs, that's where things get really tricky. This can be very, very difficult. We're lucky here at Life South that we have genotyped many thousands of donors over the past quite a few years. We have a large inventory of frozen rare cells, but if we get an antibody that we cannot provide blood for, then we're going to have to turn to some of these other methods. We're going to have to look at siblings. Do they have is there a sibling that is also negative for the high incidence antigen in question? We can put out a search, a nationwide search to the ARDP, and if all that fails, and we just cannot find an antigen negative unit that is compatible, sort of our last option is to provide an incompatible unit and perform an in vivo cross match. This is where we deliberately transfuse incompatible blood. We know that it's incompatible, but we carefully monitor the patient and observe for signs of a transfusion reaction. This is not an ideal situation, but in some cases of very high incidence antibodies, that will be the only option. Okay, any questions? If you have some, then you can hit me up at Jake Joy via Slack. Just make sure you do the little tag thing and add me, because I don't want to miss it. I'm not in the channel all the time. You can always also email me or Slack me individually. I hope you found some of this information useful, and I know there's a lot of it, but just kind of stick to the basics and those important key points, and I think that you'll have a good shot at tackling high and low incidence antibodies. Thank you.